Hello student, now in this particular video I want to answer a particular question, which is the stability of RNA molecule which is lesser than DNA molecule. So why is this? Why RNA molecule is less stable than DNA molecule? In order to answer this question, I want to request you all to see this diagram of RNA strand. Here in this particular strand, you can see the nucleotide molecules are staying together with the help of these phosphodiester bond. These two are the phosphodiester bond. With the help of this phosphodiester bond, the backbone has been formed in RNA. Why is it RNA? Because the 2 position of pentose sugar is ribose 1. It's not deoxyribose. So this is the RNA molecule, right? So this is OH. The carbon number 2 contains OH group. Now the lesser stability actually prevails in the environment. The reason behind is the presence of alkali in the environment. So in presence of alkali, the OH group of alkali is prone to attack the hydroxyl group of carbon number 2. It is a kind of nucleophilic attack. So this is this hydroxyl radical, this hydroxyl group is the nucleophile. And what happens is that once this particular OH hydroxyl group attacks nucleophilically, the nucleophilic attack of OH group lead to cause the subsequent nucleophilic attack at the center of phosphate group, and thereby these phosphodiester bond lead to be broken down. So ultimately it results in the breakdown of this particular phosphodiester bond. Now this phosphodiester bond is going to be broken down. So what happens after breaking down of this particular phosphodiester bond? Now have a look into this. After this, there is a particular substance which is known as 2,3 cyclic compound. It is a kind of, this nucleotide is a kind of cyclic derivative. So this is a cyclic 2'3' cyclic derivative compound is being formed, resulting in the shortened RNA molecule. So one nucleotide of RNA molecule has been released or removed from this particular chain. So the thing is, the random cleavage or the random degradation of this phosphodiester bond lead to cause the breakdown at several points of the RNA chain. So this particular breakdown can be done at here, this can also be done at here, and in every possible way, this breakdown process can be occurred. So thereby the RNA molecule will be degraded or chewed. Therefore, it can be said that in case of DNA molecule, which one is the deoxyribose, and H group is present over here, and there is no possibility of being attacked by this particular hydroxyl radical. So this is not possible to attack the particular nucleophilic attack cannot be done here. Therefore, ultimately this breakdown of the phosphodiester bond cannot be done. So this is the story behind this particular reason. So it has been found from this explanation, and we can understand that the stability of the RNA molecule is much less, since the alkali present in the environment, the presence of alkali lead to cause the lesser stability of RNA in compared to that of the DNA molecule. So this way we can say that the ribose sugar exhibits the lesser stability in compared to that of the deoxyribose sugar. Now, the DNA being the genetic material or the fundamental basis of life, the fundamental basis of inheritance, therefore the stability of the DNA molecule should be much higher in comparison to that of the RNA. It has been proposed that the RNA molecule was the primitive nucleic acid, and the necessity of the stability or the during evolution process or during the course of evolution, it has been the upgradation or the modification of these OH hydroxyl groups 2 prime at 2 carbon number 2 hydroxyl group at carbon number 2 has been modified or has been changed into deoxyribose, or OH has been replaced by H in case of DNA. So the DNA for the modification or gaining the better stability, the DNA molecule forms the deoxyribose sugar. So in this way we can explain that the DNA molecule is much more stable because it contains deoxyribose sugar, while in case of RNA it contains the ribose sugar. So I hope this explanation is quite understandable, and this is quite sufficient enough to address the problem or the question raised at this particular point. So I hope you have understood this particular discussion, and thank you so much for listening to me.